So good morning. Today we shall discuss on chromatography. Chromatography, in our earlier lecture, we talked about chromatography, different kinds of chromatography. Today we shall discuss a bit detail of chromatography, its principle and different kinds of chromatography. Chromatography, this word comes from a Greek word chroma. Chroma means color. And another word that means it comes from two words. One is uh, chroma, another is graphene. Chroma means color and graphene means to write. Uh, and the chromatography is the collective term for a set of laboratory techniques for the separation of mixture. Separation of mixture of compounds, different chemical substances. So different chemical substances, when they are in mixture form, uh, and if we wish to separate and purify a target compound, bioactive compounds, for example, then we use chromatography. The mixture is dissolved, initially the mixture is dissolved in a fluid called the mobile phase. That means we dissolve the mixture of the compound, for example, plant extract or microbial extract, in a solvent like ethyl acetate or acetone or methanol or any other organic solvent, and it is called mobile phase, which carries it through a structure holding another material called stationary phase. That means chromatography has two phases. One is mobile phase, where the mixture of the compounds are dissolved. Another is stationary phase, which is stationary. For example, silica gel or like that. The various constituents of the mixture travel at different speeds through the stationary phase, causing them to separate. The separation is based on differential partitioning between the mobile and stationary phase. Mobile and stationary phase that react one another, and different compounds in the mixture have different molecular weight as well as their solubility, or how to say polarity in the solvent, and their attachment to the stationary phase. So when you run the mixture through a stationary phase, that speed of the movement of the compounds are become different. As a result, we can get the separated compound. So chromatography, literally color writing, was first employed by Russian scientist Mikhail uh, Tiesvet in 1900. He continued to work with chromatography in the first decade of the 20th century, primarily for the separation of plant pigments. So chromatography was first used to separate the plant pigments. Plant contains different kind of pigments like chlorophyll, xanthophyll, or others. Chlorophyll, carotene also a plant pigment, and xanthophylls, chlorophyll, carotenes, and xanthophylls. They are, we discussed in our study on uh, phenolic compounds as well as uh, terpenoidal compounds. Since these components have different colors, green, orange, yellow respectively, they gave the technique its name. That is, as chromatography was first used to separate the coloring compounds, the color came to its name, and we called chroma. Chroma means color. Chromatography usually consists of mobile phase and stationary phase. I already mentioned. The mobile phase refers to the mixture of substances to be separated, dissolved in a liquid or gas. In most cases, we use liquid, but in some cases, for example, in case of gaseous compound, we dissolve the compound in the gas. in case of gas chromatography. The stationary phase is a porous solid mixture through which the sample contained in the mobile phase percolates. This is very important. What is stationary phase? Stationary phase is a porosity, for example, silica gel and others. They are very high, solid mixture through which the sample contained in the mobile phase percolates. Percolates means move through. The interactions between the mobile phase and stationary phase results in the separation of the compound from the mixture. Uh, the chromatographic technique is used for the separation of amino acids, proteins, carbohydrates, and secondary metabolites. So the chromatography, if we consider its application is robust. A large, or diverse compounds can be purified and separated by using chromatography. For example, secondary metabolites, amino acids, proteins, and carbohydrates. It is also used for the analysis of drugs, hormones, and vitamins, helpful for the qualitative and quantitative analysis of the complex mixture because HPLC and some other chromatographic technique they are used to quantify the compounds present in the mixture. The technique is also useful for the determination of molecular weight of proteins because some chromatography is used to determine the approximate molecular weight of a protein. The most popular chromatography is the simplest chromatography is paper chromatography, which is a technique that involves placing a small dot or line of sample solution onto a strip of chromatography paper. Chromatography paper is a bit different, and the paper is placed in a jar containing a shallow layer of solvent, organic solvent usually, and it is sealed. As the solvent rises through the paper, so paper is porous paper, so solvent can go upward to the paper. It meets the sample mixture, which starts to travel up the paper with the solvent. Let me show you. For example, this is a chromatographic tank, and this is the tank, and this is the paper, which is used for chromatography. Here, you use the mixture of the compound dissolved in a solvent, and then if you inject here a few microliters, and then if you put this paper onto a jar, in a jar containing solvent at the bottom, then what would happen? Solvent, as the paper is very porous, solvent will go upward, and it interact with the mixture of the compound, and compounds with the solvent as organic solvents is used mostly. Uh, uh, they are also moving, and different compounds have different molecular weights as well as affinity to bind to the polarity is also different, and affinity to bind to the paper different. This is why the traveling speed of different compounds are different. As a result, we can find a separation of different compounds. So by this way, we can separate the mixture of the compound. Here you see 1, 2, 3, 4, 5 compounds in the mixture, which initially we used, and they are separated. So we can understand which compound is which and what are the components in the mixture. So it moves, against the gravity. This is why molecular weight of the compound, the heavier compound moves slower than the lighter molecular weight compound. So here you can see when you put the pepper in the solvent in the tank, solvent will move faster. Compound moves depending on their chemical properties. So As compound, for example, this compound travel this part. Distance, sample travel 2.5 centimeter and solvent travel 5 centimeter. So we can calculate the speed of the compound by using a formula called RF. Distance traveled by the substance, distance traveled by the solvent. It is called RF. RF value, for example, here RF value would be 2.5 by 5, that means half. The compound under this blue color compound has RF value half, and it is an indicator of the chemical properties of the compound tested in the paper or TLC, paper chromatography or thin layer chromatography, and this is depending on the chemical properties of the compound. RF value of different compounds are different, and the RF value helps for the identification of the unknown compounds. Because if you know the RF value of certain compound, then you can easily recognize based on the RF value which compound is which. Sometimes it is rather difficult to separate a complex mixture of substances by a single run with one solvent system. This is very important point. In such a case, a second run is carried out by a different solvent system in a direction perpendicular to the first run. This is referred to as two-dimensional chromatography. So what is a two-dimensional chromatography? Sometimes when we run a mixture of compounds in a paper chromatography or thin layer chromatography. In that case, one run may not separate all the compounds. In that case, we may use a different solvent and the place the plate perpendicular to the first run. And as a result, new separation we can find, and it is called two-dimensional chromatography. For example, this one you can see. This is the first run. First run gave the separation like this way, and if you put this one at the bottom and like this way, then again run with another solvent, solvent 2, then you can find this nicely separated, uh, compounds, and it here not nicely separated, but here you see all are distantly moved, and it is called two-dimensional chromatography. And in case of paper chromatography as well as thin layer chromatography TLC, we can use this two-dimensional chromatography for better separation of the mixture of the compound. So thin layer chromatography, already I pronounced several times. Thin layer chromatography, or in short TLC, is a widely employed laboratory technique and is similar to paper chromatography. The principle is almost same. However, instead of using a stationary phase of paper, it involves a stationary phase of thin layer of adsorbent like silica gel, alumina or cellulose. So these are the adsorbents used on glass or aluminum, plate in a thin layer, and solvent runs through the, adsorbent. Compared to paper, it has the advantage of faster runs, better separation, and the choice between different adsorbents. So you can use different adsorbents, to see which adsorbent is better for separation of your mixture. Here you can see this is a thin layer plate and a solvent mixture here. Here you see the cover plate, and this is the thin layer plate, and this is the tank where we use for chromatography. We use a small thin layer of solvent mixture, and solvent moves through the this thin layer a chromatographic plate, and if you put compound, then compound runs and separate. And this is the diagrammatically we show how thin layer chromatography is done. Another chromatography is gas liquid chromatography. Gas chromatography, also sometimes known as gas liquid chromatography, is a separation technique in which the mobile phase is a gas. Instead of silica gel or cellulose or paper, here also a chemical solvent. Instead of chemical solvent, here mobile phase is the gas. So gas is used as the mobile phase. It is the method of choice for the separation of volatile substances, because volatile substances, they become a gaseous form under certain condition, or the volatile derivatives of certain non-volatile substances. The stationary phase is an inert solid material, porous materials impregnated with a non-volatile liquid. So in gas chromatography, a sample is rapidly heated and vaporized at the injection port. The sample is transported through the column by a mobile phase that is gas consisting of an inert gas. Usually inert gas is used. For example, nitrogen is an inert gas, helium is an inert gas. So sample components are separated based on their boiling point and relative affinity for the stationary phase, which is most often a viscous liquid, wax or like that within the column. The higher a component's affinity to the stationary phase, the slower it comes off the column. The components are then detected and represented as peaks on the chromatogram. So as peaks, and even gas chromatography sometimes used to mass spectrometry, so we can get even mass spectrum of the compound. So the mixture of volatile material is injected into the column along with the mobile phase. The separation of the volatile mixture is based on the partition of the components between the mobile phase and the stationary phase. Here mobile phase is gas and the stationary phase is liquid in case of gas chromatography. Hence the name gas liquid chromatography. And if this gas liquid chromatography is connected to a mass spectrometry, then it is called GCMS, gas chromatography MS. So we can even get the separate molecular weight of the separated compound. Here you can see how gas liquid chromatography is done. Here gas is provided from a gas cylinder to the system. Here is the injector. So you inject the compound dissolved in a solvent, and you inject through this here, and then it moves through the stationary phase, and due to high temperature in the oven, it becomes gaseous form, and through the gas provided, it moves, and depending on the affinity of the compound to the stationary phase, the movement of the gaseous compounds are different, and they are the first one when comes to the detector detect the compound and give a peak, and you can find the peak like this way. So gas chromatography gives the peak, but if it is connected to the mass spectrometry, then this compound again process for the mass spectrometry and give you the mass spectrum. So it is well suited for the use in the petrochemical, environmental monitoring, as well as industrial chemical fields. Gas chromatography is sensitive, rapid, and reliable. This is why it has been used in many purposes. Adsorption TLC. There is another thin layer chromatography called adsorption TLC. Adsorbents such as activated silica gel, alumina, Kaiser are used in that case. adsorption column, adsorption thin layer chromatography. Adsorption column chromatography is another chromatography. Here, column is used instead of the plate. Column chromatography is a separation technique in which the stationary bed is within a tube. A stationary bed, stationary materials are within a tube, glass tube, and column chromatography we use for a separation of higher amount of compound, because in case of TLC, in case of paper chromatography, we can only detect but we cannot recover the purified compound at higher amount. But here in column chromatography, the advantage is you can separate the compound, and here stationary bed remains within the tube. Adsorbents are packed into a column in a glass tube. This serves as the stationary phase, leaving an open unrestricted path for the mobile phase in the middle of the tube. Adsorbents such as silica gel, alumina, charcoal powder, calcium hydroxy, apatite are used as adsorbent in case of column chromatography. So if you are asked what are the adsorbents or used in the stationary phase of a column chromatography, you can name silica gel, alumina, charcoal powder, and calcium hydroxy apatite. The sample mixture in a solvent is loaded on this column. This is a glass column, that is glass tube, big tube. A tube can be different size. The individual compounds get differentially adsorbed on the adsorbent. The elution is carried out by the buffer system, which is the mobile phase. So elution, that means some solvent system we pass through the column, and due to gravity it moves toward the gravity and pass through the stationary phase, which separate the compound. The individual compounds come out of the column at different rates, which may be collected separately and identified. Here you can see the picture of column chromatography. This is the initial position. Here the adsorbent stationary phase are for example silica gel are used. Here glass wool is used so that this adsorbent cannot go out from the column. This is a glass tube. Here is a stop cork and a mixture of the compound you place here. Small amount of solvent you use to dissolve the extract, and you can inject here, place here, and then you use a solvent mixture. For example, ethyl acetate and methanol, 25:1 or like that you put here, and up to this, for example. And then when you open this stop cork, then due to gravity, solvent with the mixture of the compound start to pass through, travel through the column due to the gravitational force. And during traveling, depending on the molecular weight as well as the affinity of the compounds to the adsorbent or stationary phase, the speed of the movement of the compound toward the gravity obviously be different, and then they travel into different speed, and you can collect the eluent here in test tube, and you can find the different compound in different test tube. So you can collect 10 ml, 10 ml of each fraction, and in the fraction by using TLC you can see how many compounds are there. When you run this one, you see and the compound is moving and compound is separating like this, compound A, B plus C or C, like this way. So different colored band even we can see from the outside, and this is very important chromatographic method we use in the a chemistry lab for phytochemistry lab, or organic chemistry lab for the separation of compound. And this column chromatography is not only used to separate the mixture of compounds from the natural sources or extract of different organism, but also in the chemical reaction lots of mixtures of compounds are produced. So synthetic chemists, they also use this column chromatography for separation and purification of the compound. So another chromatographic method is called ion exchange chromatography. Ion exchange chromatography, usually referred to as ion chromatography, uses an ion exchange mechanism to separate molecules on the basis of their electrical charges. So some compounds have electrical charges. So in that case, you use ion exchange chromatography because of uh, uh, you know uh, uh, the exchanges of ion between the stationary phase and the compound helps them to separate. Ion exchange chromatography uses a charged stationary phase. So in that case, charged stationary phase is used to separate the compound, including anions, cations, amino acids, peptides, proteins. They are, they are used, these compounds, different ionic compounds as well as amino acids, peptides, and proteins are separated by the ion exchange chromatography. Here you can see how elution similarly and you can use the sand, glass, or so here you can put the ion exchange resin. So you can use the resin which exchange the ion with the compounds, and finally you can separate like the column chromatography and collect the eluent. Cation exchangers and anion exchangers are used as ion exchange resins. So ion exchange resins mostly two types: cation exchangers and anion exchangers. In conventional methods, the stationary phase is an ion exchange resin that carries a charged functional groups that interact with oppositely charged group of the compounds to retain or extract as well as to exchange the ions. As a result, the compound moves in different speed, and you can get the separation of the mixture. Gel filtration chromatography is another important chromatographic method. Size exclusion chromatography is also known as, that means gas gel filtration or permeation chromatography is also called size exclusion chromatography, and it separates molecules according to their size, shape, and molecular weight. So these three properties of the compound is considered uh, uh, in separating the molecule through GPC. It is also referred to as molecular sieving or molecular exclusion chromatography because size, shape, and molecular weight. So this chromatography used like a sieving, Chaluni like principle. Smaller molecules are able to enter the pores of the media, and therefore molecules are trapped and removed from the flow of the mobile phase. However, molecules that are larger than the average pore size of the packing are excluded and thus suffer essentially no retention. Such species are the first to be eluted. So it is using just like sieve. So if molecule is larger than the porous pores, then it cannot pass through. This is how the molecules are separated. It is generally a low resolution of chromatographic technique, and thus it is often reserved for the final polishing step of the purification. It is also useful for determining the tertiary structure and quaternary structure of purified proteins, especially since it can be carried out under native solution condition. And obviously This chromatography is used for final purification or final step of the purification of the protein or large molecules. Gel filtration chromatography, here you can see how it works. You can mixture of the target and small proteins and chromatographic graphic column. This is the column and tubes for collecting the proteins after separation. So you see how sieving and eluent are coming. So different eluent depending on the molecular weight as well as their size and shape. Because protein has size, shape, and three-dimensional structure. So gel used to separate different proteins through this chromatography. Affinity chromatography is based on selective non-covalent interaction between an analyte and specific molecules referred to as ligands. The affinity chromatography is also used to separate the big molecules like protein. The immobilized ligands act as molecular fish hooks and selectively pick up desired proteins while the remaining protein pass through the column. So here you see affinity, some compounds they show higher affinity to pick the bind to the ligand, and they are stack here, and those show less affinity, they pass through the column. So ligand hook like this way. The compound here you see the ligand help certain compound has higher affinity to bind with the ligand, and they move slower, and other proteins then pass through the column. The most important and last topic is high performance liquid chromatography. High performance liquid chromatography at BSMRU we have in the central lab. High performance liquid chromatography, it is a liquid chromatography, is a separation of technique in which the mobile phase is a liquid. Liquid chromatography can be carried out either in a column or a plate. Present-day liquid chromatography that generally utilizes very small packing particles and a relatively high pressure is referred to as high performance liquid chromatography. So HPLC is a liquid chromatography, and here a high pressure is used to separate the compound, and small amount of compound can be purified, separated and purified by using HPLC. So HPLC is used for the final stage of purification of the compound. We cannot use the crude mixture of the plant extract or like that. Since the chromatographic techniques are slow and time consuming, hence the separation can be greatly improved by using high pressure in the range of 5,000 to 10,000 psi. Hence, this technique is also referred to as high pressure liquid chromatography. So high performance liquid chromatography because its performance for separation and purification of compound is high, and it also use high pressure for separation of the compound. So we can call it high pressure liquid chromatography. In HPLC, the sample is forced by a liquid at high pressure. The mobile phase, we can use an organic solvent like ethyl acetate and methanol mixture or like that through a column that is packed with a stationary phase composed of irregularly or spherically shaped particles. So in that case, stationary phase like column chromatography similar. Column is used here, packed with irregularly or spherically shaped particles. The interaction between the mobile and stationary phase leads to the separation of the mixture. Here you can see the high pressure liquid chromatography. We can use different solvent, for example, here ethyl acetate, solvent 1, solvent 2, for example, methanol. So they can mix together. You can regulate in which ratio they can mix together, and high pressure they move this way, and there is a vacuum pump work, so solvent moves through this way. And here there is a pre-column, so this solvent mixture pass through this pre-column and coming through this way. And here you see sample injection port. So sample you inject here, dissolving in small amount of a chemical solvent, and mixture of the compound, 2, 3, or 4 compounds mixed together. And then you see solvent is coming here, and solvent and this sample mixture, they through the pressure, they moves through this column packed with, I told you, different porous materials, and this analytical column separates the compounds depending on their molecular weight, their affinity towards the stationary phase, as well as other properties, and then it moves here and finally give you the peak. There is a detector here, and you can get the peak here, and certain compound, when it is coming, a peak starts like this way and then go down. That means this compound is over, then second compound comes, or third compound. So depending on the chromatogram, you can understand which compound is coming out, and you can collect the eluent. And when peak goes to ground, then you can stop and collect in different tube for different compounds. So we can get the almost very high level of purified compound by using high performance chromatography. And this is sometimes connected to the mass spectrometry. In that case, mass spectrum data you can find. So HPLC MS is now a modern equipment for getting purified compound as well as its mass spectrometry. So this is all about for today's lecture. Please ask some questions so that we can discuss more.
স্যার আসসালামু আলাইকুম।
Walaikum
জিনান
salam.
স্যার।
Yeah, Jinan.
স্যার স্যার uh, is the molecular weight of protein can only be determined by gel filtration chromatography, or we can use other chromatographical process to identify the molecular weight of protein?
A molecular weight of protein. Yes, we can use gel filtration for separation and purification of the protein, but we can guess, the how to say, approximate molecular weight of the protein by gel filtration chromatography. But other chromatography, for example, silica gel chromatography, paper chromatography, and thin layer chromatography, we cannot handle protein or like that. Protein is a very big molecule. This is why we need, other method like electrophoresis. Electrophoresis is another method we shall discuss later on. We use for understanding the molecular weight of protein, nucleic acids like DNA, RNA. Mostly electrophoresis is used for determination of the molecular weight of protein and big molecules. And chromatography, today I discussed, you can get an approximate idea of the molecular weight by using gel filtration chromatography of a protein, but not too precise. Thank you.
Thank you, sir.
You're welcome. Any other? Do anyone have any more question? Have you started, find your paper from the, journal? And it is important because if you start now and every day, if you browse a little bit, maybe you need a bit time, because lots of paper uh, are published in international journals, but many of them may not, link to your course, bioactive natural products, as well as some of them are maybe very complicated. So in that case, you need to search several journals and several volumes of the volumes to find the paper. Best idea is to use keyword if you love terpenoid, and then put bioactive terpenoid and browse and see whether there are any papers published or not. You can use bioactive flavonoid or bioactive alkaloids or like that. So browsing your chosen keyword, you can find the paper. But obviously, you have to spend time to find a suitable paper for your seminar work, as well as to learn the new discovery published in the field of bioactive natural products. And every day some new compounds are being discovered by the scientists somewhere in the world, and they are publishing in different journals.
স্যার আসসালামু আলাইকুম স্যার, সাবরিনা
Yes, Sabrina.
স্যার স্যার যদি আমরা প্ল্যান্ট থেকে কোনো কিছু এক্সট্রাক্ট করতে চাই, তাহলে স্যার সেটা পিউরিফিকেশনের জন্য স্যার কোন ক্রোমাটোগ্রাফিটা ভালো হবে আমাদের?
In case of plant, if you want to discover secondary metabolites from plant, in that case we usually use first time the thin layer chromatography to see the how many compounds in the mixture, as well as to know the solvent system, which are system which is the best for separating the mixture of the compound. And same solvent system then we use in the column chromatography for the purification of the compound. So thin layer chromatography, and then column chromatography, big column for separation of the large amount of compounds. And then when you get semi-purified compound, for example, 2 compounds in a mixture, after the column chromatography, sometimes column chromatography you need to do several times, and then you can use the HPLC. So only single chromatography may not give you a purified compound. So in case of plant secondary metabolites, if you want to discover a new bioactive compounds, we use the bioassay guided fractionation by using column chromatography. And when you get the fraction nearly pure and contains your bioactive compound, then you can use the HPLC for final purification. After purification, you must need to do the bioassay again, and then you get the purified compound. And I think this is the usual way we use for separation and purification of the compounds from the plant as well as the microorganism. Any more question? Very nice question, Sabrina. Any more question? Please ask some question. Then you would be, get more clarification about the chromatography and their practical use. Yes.
Assalamu alaikum, sir.
Wa alaikum salam, Mujahid.
Sir, if I find a flower with a beautiful fragrance, and I will try to differentiate this fragrance and try to elucidate the chemical structure of the fragrance, and what method I could use?
This is an interesting question. When you find a flower which has a very attractive fragrance or aroma, and if you want to target to purify that compound, which chromatographic method you use. In that case, I suggest gas chromatography, because gas chromatography can easily handle the volatile compounds, and most of the fragrance compound have already been discovered by the scientists by using gas chromatography. So you can find in the database uh, the gas chromatographic peak and other information. So you after doing the gas chromatography, you can compare as well as nowadays gas chromatography is connected to the mass spectrometry. So if you get the gas chromatogram, uh, the which peaks are at which point the peaks are coming of the particular compound, as well as its mass spectrometry, then you can easily make a conclusion what compound is it. But if you want to uh, uh, get the purified compound at larger amount, you can also use silica gel, column chromatography, or even HPLC. HPLC, because fragrant compound having fragrance can be dissolved in organic solvent, and then you can use HPLC for purification of the compound. That is, if you may find it volatile oil or like that, but trapping of the oil in some adsorbent is important. Otherwise, the fragrance may move to the air, and you may lose the compound. And gas volatile compound, discovery of volatile compound requires special skill, as well as special care during the you know purification process, as well as getting the compound in the solvent. Thank you very much. Any other question? Any more question from you? No? If no question, I would like to thank you for attending this class amid the pandemic coronavirus. And please take care of yourself as well as your family members, so that you can keep you free from the coronavirus. It is a highly contagious disease, and more than a lakh people in our country contaminated, and many people died. So every day we are getting bad news through the television. So I wish you a safe stay and a safe and healthy stay during this pandemic period. And I hope you are enjoying bioactive natural products, this course. And if you find any question at any point of your learning process, don't hesitate to ask your question through the Facebook group, because it is a nice virtual classroom for you. Thank you very much.